All right, hello students. Welcome back again to MOOC course Introduction to Proteomics. As well as I think we have some students joining from course Interactomics. This live session is essentially to teach you something about one of the type of mass spec, which is MALDI-TOF. So in the basic lectures, I think you are getting an idea that how proteomic technologies are evolving, in which way we can type of mass spectrometry instruments for doing proteomic analysis. Of course, it may be a little still early after just week one for you to be on top of knowing these technologies. But we are keep showing you the lab views and some of the actual instruments that what one could do out of them. So today we thought to show you a demonstration for MALDI-TOF instruments. Now, just kind of very briefly, some of the basics of mass spec. So in any mass spec, you have one ionization source, you have mass analyzers, and you have detector. So when you are terming any kind of mass spec configuration, right now we are talking about MALDI-TOF. So we have MALDI, which is one of the ionization source, and sometimes you may have here heard ESI or electron spray ionization, right? That is another type of ionization source. So you have your molecules of interest to analyze, especially proteins or peptides, and now you want to ionize them so that those ions could move inside the mass analyzer. In this case, we have mass analyzer, which is TOF, time of flight tube. And you can have different type of mass analyzers, depending on the type of principle which you want to use to separate those ions. So right now we are using two different type of TOF tubes, TOF1 and TOF2. Sometimes you may have configuration which is having QTOF, quadrupoles and time of flight configuration. So in today's demonstration, you are going to see the MALDI-TOF instrument, which is one of the hybrid MS configuration, and that you can say the MS technology. Here you have MALDI as ionization source, two of the TOF analyzers as an ionization mass analyzers. So when you want to analyze any of your protein samples in MALDI, then first of all you have to crystallize those, because you have to do the laser desorption ionization. So to do that, you are adding certain matrices, and then you are making the solid crystals, and those crystals are then put it inside this instrument. So right now I'm standing in front of this MALDI-TOF instrument, in which we have this particular plate, this area where I can actually take the camera to you and show you a little bit in more detail. So here, we can insert the plate. Can I get a plate? All right, so well, I'm trying to just kind of have a little bit hands-on and show you a little more detail about the instrument. So this is a MALDI plate on which you can put your samples of interest, right? And that you're going to see in much more detail. And then you're going to place it inside this particular slot to insert the plate, right? And then after that, everything is pretty much software operated. So let me go back to my position. Yeah, so entire thing, first you are starting with the sample preparation. When you are using your protein of interest, adding the matrix, mixing them well, then you want to place those on the plate, MALDI plate, which is sometimes 384 well plate or 96 well plate. And on these plate, then you are having your protein of interest, which you want to analyze. You are inserting inside this instrument, and then you are doing a laser bombardment on that. And then if your crystals have been formed properly, then probably you can use those information to analyze in the mass analyzers. Sometimes you want to do only PMF, peptide mass fingerprint analysis, or sometimes you want to do MS analysis. If your intention is just to know the mass of a given compound or a given molecule, then even PMF can be provide you good information. But if you want to obtain the sequence level information, then you should do the MS analysis. So while right now mass spectrometry technology is undergoing a lot of revolution, a lot of changes are happening, and you might be hearing that many type of LC-MS platforms are becoming much more robust for doing a lot of proteomic analysis. But I must say that I'm totally not convinced with those statements, just because each instrument has their own unique capability, and it offers you certain very unique application which you cannot obtain from other instruments. So for example, if your interest is to identify, just imagine from the 2D electrophoresis or from SDS page gel, you have obtained lot of bands and the spots of interest, and you want to just identify all of those proteins very quickly. Now, if you want to do that, you're in the multi-plate, you have option to spot, maybe hundreds of a spot. And then all of those you can put in a plate, you can leave it overnight unattended, and just define to the software that it has to do analysis and laser bombardment on all of these spots. So unattended manner, in a very fast way, you can acquire spectra from those, do MSMS analysis. Even for MSMS, you can define that how many peaks has to be selected, and it can do all auto bombardment, and as a result, you can obtain all the spectra pretty much unattended form. And it takes very little time for doing this entire high throughput analysis. So maybe in a day time, you can have protein identification for hundreds of spots, which is not possible in LC-MSMS based approach. In LC-MSMS, you are separating all of those compounds based on their different principles using liquid chromatography, which takes much longer time to separate the peptides. So then therefore you are spending longer time, at least an hour or so or more, for even each of the identification. So just you have to weigh the pros and cons for different technologies. For example, different type of mass spec configurations, all of them are actually having some value added information which you can obtain. In this case, I prefer MALDI for some very quick analysis. If I want to just know the mass of a given compound, we have purified a protein, and I just want to know that what is the molecular weight of the purified protein. Is it matching to my known or expected molecular mass? And then if that is the case, then probably I'm very happy that this is probably right. I don't need to do anything else from any other sophisticated mass spec instruments. MALDI-TOF can do that part. Or if I want to know the sequence of a an unknown protein, and I just want to identify those protein, I think this particular application can be done very well here. But when you have very complex mixture and you want to analyze those in the in-solution form, and you want to really quantify them well, then I think that then it becomes much more specialized application. That's where probably LC-MSMS based ESI, QTOF or Orbitrap platforms are much more robust. So, I think what I really want to involve you in some sort of discussion and gauge the interest to find out what are the pros and cons of different type of instrument platforms, different technologies. Of course, each day we cannot cover every technology, but today our intention is to talk to you about just one of the instruments here, which is MALDI-TOF. And what we have, planned for you, a little bit theoretical part, some basics part which we are going to cover. Along with that, we're also going to show you some hands-on how to take the plate and put the matrix and samples, how to prepare the plate properly, and then what you can do to insert the plate and then analyze using a specialized software which we have to do this kind of analysis using MALDI-TOF mass spec. So let me introduce with me one of the research associate, Dr. Aishwarya Rao. So Dr. Aishwarya Rao, she is an application scientist. She is working on the handling MALDI-TOF based applications at IIT Bombay. She's going to talk to you in much more detail about some of the basics of MALDI-TOF, and then she is going to walk you through one of the demonstration session. I also have with me a teaching assistant. Please come here. He's Mr. Vipin. He's a senior PhD student. He is pretty much involved in this course as the official TA for the course. He is going to show you the hands-on part of how to prepare the samples. So again, in the spirit of you to become much more comfortable about knowing proteomics technologies and what you can get out of them, I think we are planning for each week certain hands-on session and live demonstrations. I'm sure many of you will be able to make good use of them. So please, let me continue the session with them, Dr. Aishwarya and Vipin, to take you to the actual workflow of doing MALDI-TOF based sample analysis, and then post that, then we can take more of your queries and comments. So let me thank you here, and then I will hand over this thing to Dr. Aishwarya. Please.
Thank you, Professor Srivastava. So good evening to all the participants who are there online. Like Professor Sanjeeva just now said, we'll be handling the MALDI-TOF instrument today. Details of this would be discussed. So this instrument, like he rightly mentioned, is used mostly for the analysis of proteins, polymers, nucleic acids, carbohydrates, and a lot of other samples. Majorly, the application is to find the molecular weight of these samples. And in the case of proteins, we can also try and identify the proteins, unknown proteins that are present in the gel pieces. So saying that, we would now like to go on to the little presentation that we've made for you. So MALDI-TOF is a type of mass spectrometer that is, like I said, for the analysis of various molecular, to find the molecular weight of various compounds. So this, before we move on to the MALDI-TOF, we would first like to talk about the different spectrometer. Any mass spectrometer for that matter generally has three basic parts. One is an ionization source. The second part is a mass analyzer, and the third part is the detector. The ionization source in case of biological samples could be either electron spray ionization or a nitrogen laser, which is used in the matrix assisted laser desorption ionization method. The mass analyzer in biological sample analysis is usually either a quadrupole, time of flight, or ion trap. The sensitivity of the instrument is really determined by this part of the mass spectrometer, that is the mass analyzer. There could be just one analyzer or two or more analyzers put in tandem. So there could be a quadrupole followed by a time of flight and an ion trap to increase the sensitivity of the instrument, or like in today's case, there could be two time of flight or there could be two quadrupoles as the case may be. The detector is usually an electron multiplier and is also one of the core parts of this instrument. This technology was, which is the matrix-assisted laser desorption ionization, was developed in the mid-1980s by two German scientists, Michael Carus, who was a postdoc working in Franz Hellenkamp's lab. They were basically chemists who developed this technique for soft ionization technique for the study of few chemical compounds, which they did not want to completely break down with the help of other ionization techniques that were prevalent at that time. But it took a long time for this technique to be used for biological samples. And it was Koichi Tanaka who got a Nobel Prize for showing that this particular technique could be used for biological samples in the year 2002. So the basic components of the MALDI-TOF instrument are the ionization source. Over here, we use a modified nitrogen laser. The mass analyzer is a time of flight, which is actually a vacuum tube through which the ions that are generated move and are separated on the basis of their size by mass by charge ratio. And finally, the detector. In this particular technology, the matrix is of primary importance. There are different types of matrices which are used as per the requirement of the sample. What this matrix does is that it first co-crystallizes with the sample, and when the laser beam strikes these crystals, the matrix will absorb all the energy from the laser beam, will convert into an ionic state, and as this process is going on, the energy will then be transferred by the matrix ions to the sample molecules, which in turn will get ionized. So it is imperative that the matrix and the sample molecules are able to interact and this energy transfer takes place in a proper way. There are different type of matrices that are generally used for MALDI, depending, like I said, on the type of sample. If there are peptides, we usually use a matrix HCCA. This crystallizes in different way, and a picture of it is right now on your screen. For proteins which have molecular weight more than 5 kilodalton, cinnabic acid is generally used, while for nucleic acids, 3-hydroxypicolinic acid is used. The 2,5-dihydroxybenzoic acid is used for the study of glycans, small molecules like polymers, and other substances. So these are just a few of the examples of matrices that I use. There are many more depending on the type of sample There could even be a combination of different matrices. Some salts could be added in. Some other phosphorus containing compounds could be added to enhance the capability of the matrix. Now coming to the instrument per se. So to the left of the slide is an image of the autoflex speed machine that we have here at IIT. Professor Sanjeeva showed you. There is a tray that comes out, a sample loading tray that comes out, which has the target plate. So from here, my colleague Vipin will be showing you how samples are loaded onto the target plate and how it is placed in the instrument.
Yeah, hello. So basically, this is a multi-target plate, which is a ground steel plate, and it having a 384 well, where we can spot at a time 380 spots of the samples. So it is very fast. So we can analyze all samples at a one time. So here I'm going to like how to spot the sample. So basically, the Aishwarya already explained about the matrix and the sample, like which matrix are suitable for which type of samples. So basically I have today digested proteins that are basically peptides. So basically we take the equal volume of the peptides, that is sample. So I am taking here 2 microliter of my peptides. Then I will keep in the appendo. Then I will take 2 microliter of the matrix. So the matrix is for the peptide is HCCA matrix. So I'll take 2 microliter. So basically I'm taking equal volume of the matrix and the samples. Then I will dissolve properly. Then I will take 2 microliter of the samples and then I will spot. Now, so we spotted only one sample. Now we have to wait for 10 to 15 minutes, and then once the samples will be get dry, so then we'll put in our instrument MALDI-TOF, and then we can do the analysis. So basically now I will show also like how to put those like plate in the MALDI. So I put the plate here, and there is actually you can see the barcode is also there. So this barcode should be this side. So here you can see now the plate is going inside. Now like plate once inside, so it will start to create the vacuum. Then we can start the analysis. So the instrument will be controlled by the flex control system, and then we will acquire the data. So after acquiring the data, we have to do analysis of data by using flex analysis software. So which we are just going, will show now.
So thank you, Vipin. So what he has right now shown us is one way of loading the sample, which is the direct droplet method, wherein the sample and the matrix are first combined in a different tube externally. They are mixed together in equal proportions, and then they are loaded onto the plate. There are different ways of loading samples. There is another way which is called the sandwich method, and in this method, a layer of the matrix is first applied to the ground steel plate. Once that dries, on top of that, the sample is added, and when the spot for the sample dries, another layer of matrix is given on top. So depending on the sample, the loading technique, the matrix, all these are decided. So now, as we continue with our presentation, we have, once the plate is loaded into the instrument, we analyze the sample with the help of flex control. The flex control is the software that will take all the data and The data is then transferred to the next software for analysis, which is called flex analysis. So we would like to give you a small demo of one of the analysis that was performed a while ago. And before we do that, let's see what exactly happens inside the instrument. So let's say we take a vertical cross section of the instrument. That is how it would look on the left-hand side of the screen. The plate is right at the bottom of this instrument right now. The laser beam, which is shown in red, will strike the spot which has been, strike the sample which has currently been spotted onto the plate. The energy from this laser beam will be absorbed by the matrix molecules and get converted to ions. The ions, this will then transfer energy to the sample molecules so that they get converted to the ions in their time. Then you can see two other rings right above where the target plate has just now been kept. There would be an electron cloud that is first concentrated between these two plates. And in this region, there is a transfer of charge that happens from the matrix ions to the sample ions. So in case of MALDI, generally one positive charge is transferred from matrix ions to sample ions. Once this is done, we can select for the size of the ions which have to enter into the top tube. So as these ions now move into the vacuum tube, they get separated on the basis of their mass by charge ratios. So the smaller ions move faster in this vacuum tube, and the heavier ions or the bigger ions would move slowly. As they move vertically upwards, they are detected with the help of a detector, a small camera-like structure which is present at the top of the vertical cross-section. So if the samples are more than 5 kilodaltons, if the expected molecular weight is more than 5 kilodaltons, then the ions that are generated are big enough to be separated in this path length and can easily be detected by the detector at the top of the instrument. This mode of detection is therefore called the linear mode. But sometimes if the samples have got a very low molecular weight, the ions that are generated um, are very small, and therefore this path length is not enough to resolve them properly. For this reason, the instrument is supplied with a lens, which is depicted as a yellow series of lenses over here, and the ions are then deflected along another path and then detected by a second detector which is present very close to the point of entry of these ions into the vacuum tube. And this point is called the, here there is another detector placed over here, and now since these are the reflected ions that are being detected, the detector is called as the reflectron detector, and the mode of detection is called the reflectron mode. So this is better exemplified in this particular slide. This would be the linear mode of detection, detected with the help of this detector at the top of the instrument. And for smaller molecules, we have the reflectron detector over here. Once the detectors detect these ions, depending on the intensity of the ions that are hitting the detector, we have different intensities that are recorded. So the y-axis of the graph will give you the intensity of that particular ion, while the x-axis will give you the n by z ratio. So like I said, the Flex control software is the one that is used for acquiring the data. The Flex analysis is used for the analysis of the data. And there is a third software, which is called BioTools. This is generally used when the MSMS mode is being applied in the instrument and if the identification of proteins is to be done. We will now have a brief demo of the Flex analysis.
So here basically now, after acquiring the data, we'll go through the analysis. So now here you can see the flex analysis software. So I opened two files. So here you can see there are peak. Here you can see, right? So basically now there is one experiment where they want to see the shift in the mass of the protein. So here there are two peaks. So we can see we can label the mass list also, and then here you.
Right. So in this particular experiment, we had a user coming in with a normal protein and a modified form of the same protein that was being done in the lab. So in the above screen, we have the normal protein, while the lower spectrum is for the modified protein. Though the spectrum looks very similar for both the proteins, when we found out the molecular weight with the help of mass list for both of them, we found there was a slight change. Now, if I expand this region, so if I expand this region over here, we can see that the molecular weight of the normal protein is coming around 29.5 kilodaltons, while the one for the modified is around 30 kilodaltons. This could be better represented if we overlay these two spectra. So once we overlay them and we expand the region of interest, it is very clear that there is a definite shift in the molecular weight, which means the modification that was being tried is now successful. So this is just one way in which we can use MALDI to find and to represent how the changes occur in a particular molecule. If there are some dyes that are attached to a protein, or whether there is some mutation that is happening, these studies can be taken up very well in MALDI. So this is just a one slide overview of all the applications that happen in MALDI. Like we've already discussed, the molecular weight of different proteins can be determined. The nucleic acids can also be determined, whether they are synthesized nucleic acids or they are naturally occurring. Any changes in the molecular weight of these acids, nucleic acids, are also seen. Polymer characterization is another major application that is for which MALDI is used. A lot of people come in for N-group analysis, for molecular weight determination of their polymers, and other studies. Sugars can also be analyzed using this type of technique, and the identification of proteins from gel pieces can be done with the help of peptide mass fingerprinting. So this was a brief overview of the MALDI applications, and I hand over the session back to Professor Sanjeeva.
All right, so I'm sure you got some glimpse of what one could do using MALDI-TOF based mass spec instruments. It's pretty straightforward. In fact, you have to just choose the right type of matrix, and you need to do some basic skill set to prepare the sample so that the crystallization becomes proper. And crystal formation, I must say, is a kind of success for your experiment here. If you can make good crystals, a distinct one, and then you have the proper laser bombardment on that, then you can obtain a good MS spectrum based on that. Sometimes you have to optimize many parameters to really acquire the right spectrum. And then, of course, as Dr. already mentioned to you that if you're analyzing the peptide, these kind of complex samples, then probably you need to use reflectron mode so that you can increase the path length of the overall how much these ions can travel in the mass analyzer inside the field. So we'll not go in much more detail right now. I think this was a short interactive session to just convey you one of the type of mass spec and what kind of application it can do. I'm following up some of your queries on board. If you just think about, I think one question I saw that Western blot can also give you the region, or in fact, when you can also see the exact molecular weight of a given compound, right? So then why need to do MALDI? So just imagine that for any protein, if you want to purify, or if you want to really have the Western blot, you may not have antibodies available on one hand. And that thing you can do for any given protein. When you want to do Western blot based experiment, you can do for any given compound, any given protein. On the other hand, when you want to do something in a very high throughput manner, just imagine one Western blot experiment will take you 2 days time, 1.5 days to obtain a right band, right size. Then you can here do things much faster way. Then, as I said, the major advantage here is that you can do for any compound whether you have antibodies available or not, right? And then think about the different type of application. I think some of you have asked, can you identify and get a sequence out of the MALDI-TOF based instruments? Yes, you can get, because when we are using a TOF or any MS platform, then you are able to identify for each given MS spectrum, you are further fragmenting them using collision energy, and then you are able to derive the P and Y ions. Based on that, you can obtain the peptide sequence. So of course, you can use MALDI-TOF to identify the protein based on the peptide sequences which can be generated out of it. So these are some of the interesting queries which I think as you go along in the course, it will become much clearer to you that when we are going to also share some data and a spectrum to you, that what happens from the MS to MS, in between that you have to use some collision energy to fragment those ions, and how these fragmented ion pattern looks like, and then how you can interpret those data based on the V and Y ion pattern. I think some of these things will be interesting follow-ups for the weeks to come. So I'm sure that now you are finding the course a little more enjoyable. You are at least getting some interest, and these kind of lab sessions will really give you the first-hand feel of what kind of experiments you can do using these kind of sophisticated instrument platforms. So I'm going to stop the session here right now. Of course, please feel free to put more queries and more questions which you may have directly to us. We will reply back to you. Additionally, I would like to again announce that as I mentioned in my previous introduction as well, that the following week or 10 days from now, we are going to conduct target Proteomics workshop and symposium. Some of the topper assigned people who will be getting good marks, some assignments, we are going to give them opportunity to come here and meet us directly and do some of these experiments here and attend the symposium. Additionally, I think if some of you are already interested, please do let us know. We can see how best we can try to accommodate you here, because as you can see, there are many interesting technologies and platforms available. We have many good foreign experts coming in here to train you and teach you these kind of technologies. So we would like to have your the first interaction with them. But even otherwise, certain sessions we are going to also have the live streams like this, so that you can listen to those talks, you can attend those sessions, and you can definitely try to get much more knowledge from these scientists. So thank you so much. Now we'll continue these kind of interactive sessions again and inform you ahead of time. Thank you. I'm closing the sessions now.